So LOPIT stands for Localization of Organelle Proteins by Isotope Tagging. So why do we want LOPIT? Being able to know protein subcellular localization helps you elucidate the protein's function. And so LOPIT is pretty important for this. So just an overview, but I'll explain this more in the following slides. LOPIT uses an iodixonal gradient, and you use this because it reproducibly forms the density gradients more reproducibly than sucrose. And essentially in LOPIT, you have organelle partial separation using density gradient centrifugation, followed by a protein distribution analysis using ICATs and mass spectrometry, which I'll explain more later. And multivariate data analysis groups novel proteins of interest according to the known distributions, and that's how it gives you the subcellular localization. So the main advantages of LOPIT are that it's well suited to examine proteins with multiple localizations, and this is in contrast to proximity tagging methods. LOPIT is not dependent on pure organelle fractions, and this is its main advantage. It uses analytical centrifugation to produce a protein distribution. And you'll see what I mean in a minute. So yeah, this means that LOPIT can be used to discriminate organelles of similar density. Because these organelles would be difficult to purify. So yeah, so by comparing the distributions of novel proteins to known organelle residents using multivariate data analysis techniques PCA and PLS-DEA, partial least squares discriminant analysis, you can get the subcellular localization of novel proteins. So when you have your sample, you do density gradient centrifugation, and you split it into roughly 12 fractions. And so if you take this protein, for example, you can see that this protein is spread across fractions 3, 4, 5 until 12. But then its concentration changes as you go along the fraction. And this is a Golgi protein. Contrastingly, ER proteins will have a different distribution across the fraction. So this AT-Sec12 protein, it's high in fraction 2, 3, 4, and then tails off. So what this LOPIT method does is they use ICATs, and ICATs are basically isotope-coded affinity tags, and all it does is it quantifies the relative protein levels between your two mass spec samples. So if you label, for example, fraction 3 with a light ICAT, and then fraction 6 with a heavy ICAT, you can see over here that your light ICAT, you'll have a higher quantification signal of your light ICAT and a lower one of the heavy one. And it also corresponds to the bands over here. So yeah, so there are 12 fractions, and then you do 6 pairwise comparisons, and I'll show you for what later. So yeah, so you've done this and you've done this. You have this data, you now want to assign protein subcellular localization. So you use PCA for this. And so yeah, so PCA clusters proteins with similar distributions. And so if you look at this graph over here, the squares correspond, so the filled in squares correspond to ER proteins. So if you run all this data through PCA, you get this plot basically. and the triangles correspond to Golgi proteins, and this diamond over here is plasma membrane. And so if you want to predict the localization, this protein subcellular localization of a novel protein, you can then predict its location via the PCA plot. So the unfilled squares represent, so this is like a validation graph. The unfilled square is in this cluster over here. And so you can predict that this protein is an ER resident protein, and that is the case, basically. So the authors of this paper were just trying to show that their method works. So there are two. So this is LOPIT. However, LOPIT was improved in like 2018 or something like that. And there are two variants of LOPIT. There's hyperLOPIT and there's LOPIT-DC. So hyperLOPIT is hyperplex LOPIT, and it just has a bunch of improvements, and it achieves high resolution organelle separation, but it's time costly and resource intensive. LOPIT-DC, on the other hand, is less resource intensive and less time costly. And the difference between these two is that hyperLOPIT uses equilibrium density gradient ultracentrifugation, whereas LOPIT-DC uses differential ultracentrifugation. And if you're unfamiliar with these two types of centrifugation, I'll explain in the next slide. So before I do that, I just want to mention that the cell lysis stage to produce your homogenates, this is pretty critical because insufficient lysis gives you reduced protein yields, while excessive lysis will release the contents of the organelle and you won't get accurate data. So equilibrium density gradient centrifugation. So these two, the difference between these two types of centrifugation is that density gradient centrifugation uses a gel, or one sec. So it uses a solution or gel through which the sample passes. And so if you look over here, you basically have a gel, like a sucrose gradient or whatever other gradient. So like the very first slide, it was a iodixanol gradient. So yeah, and you basically get fractions. So that's equilibrium density gradient centrifugation. Differential centrifugation is when you only get two fractions when you centrifugate. So you get the supernatant, and then you get the pellet. So differential centrifugation, you get one pellet that sediments at the bottom, and you just get a supernatant. And then you pass this to another, you can centrifugate supernatant here and then repellet it. And that's, so that's the difference between these two types of centrifugation. And then density gradient centrifugation can be further subclassified. I'm not going to go into that. You can read it if you want to. And yeah, the main difference is because you're fractionating with, in here, because you're fractionating with a density graded solution, you only use one centrifugation speed, whereas here you use successively increasing centrifugation speeds. So so hyperLOPIT and LOPIT-DC, they both use multiplex TMT labeling, and this is to reduce mass spectrometry run and analysis time, and also the cost. And it also reduces the technical variability and lets you have all the subcellular fraction analysis in a single run. So multiplex, if you're unfamiliar with multiplex TMT labeling, so these are isobaric mass tags, which just means isobaric just means molecules of the same mass, but they yield reporter ions of different masses after the tags fragment. So you may have heard of iTrack. So TMT and iTrack are pretty much have the same principle. They just have different chemical structures. So here I've just put a picture of iTrack. So you have a reporter group, a balance group, and these two always add up to the same mass, like over here, 145. But then the balance group and reporter group, they change. So you get 114, 115, 116, 117 Daltons. Sorry, kilodaltons. Is it kilo? No, just Daltons. Yeah. So iTracks and TMTs are pretty much this, but the important thing is what this lets you do is this lets you quantify the relative intensities. So for example, if you can see over here that you can see the relative abundances of each reporter ion, and then that lets you see the relative abundances of peptides, because these TMT and iTrack tags, they label peptides that are produced by protein digestion. So finally, the data analysis part is pretty similar to the original LOPIT. So you use the tags to produce the abundance versus fraction graphs. So as you see here, the abundances of the different proteins in different organelles change with the fraction, which is really similar, which is pretty much the same principle as this over here. And then this can get converted to principal component analysis plots. And then let's say you want to predict the localization of a novel protein, and you see that your novel protein, for example, lands, let's say lands over here or something, your novel protein. Then this is clustered around like nucleus chromatin, so you can predict that this protein is localized in like the nucleus. So yeah, thanks for watching. These are the two papers. I will link them in the description. And yeah, thanks for watching.